So immune system, continued. All right, so we've been talking about specific or adaptive immunity. So I just want to very briefly just reiterate some of the defining qualities of specific adaptive defenses. And what are the characteristics that define specific immunity?
Antigen specific.
Exactly, antigen specific. It's going to be created and then look for, it's created by some antigen presenting cell who's got a particular antigen that it has got from something it ate that it's presenting, and then all of the defenses that are created in response to that are going to be against that particular antigen. You know, in particular, we just saw antibody mediated, where the plasma cells are making all these antibodies that will specifically bind to that antigen, you know, and then target that for destruction. Awesome. What's another defining quality of specific immunity?
Memory.
Has memory. So again, that's super important. It means once you've seen it once, your body's going to be able to ramp up a very quick and strong response if you ever encounter it again. So you'll likely not get sick at all. Again, that's what immunity is about. That's how vaccines work. And then the last thing we talked about was that it was systemic. Like wherever the pathogen or whatever first is encountered in your body, it doesn't matter. You end up making all of these defenses that occur across everywhere in your body. And then so we looked at the two branches of specific immunity, which was the antibody mediated which we also called humoral. And then I mentioned, but we didn't talk about specifically yet, cell-mediated. So kind of where we left things was we kind of talked more in detail about antibody-mediated immunity, where you had the B cells, B cells which there's one particular B cell with a receptor that just happens to be the perfect match for the antigen that's being presented by the antigen presenting cell. Then we went through that whole clonal selection where we took the chosen B cell with the chosen receptor, and we basically ramped up production of these plasma cells that made antibodies that could bind to that particular antigen. And what happens when an antibody binds the actual antigen? How does that actually end up taking out the target? If
they just block the binding sites.
Right. So we talked about there was neutralization, that idea that you could just kind of block binding sites on the target so it can't actually do what it needs to do. Kind of can the analogy like it's sticking on my eyes, I can't see, or it's sticking all over my hands, I can't actually use my fingers anymore. What's another way that antibody binding leads to the destruction of the target?
It can activate the complement system.
Exactly. It activates that whole complement cascade. And once that happens, all those plasma proteins ultimately result in those membrane attack complexes, big holes opening up in the membrane. It opsonizes the target, makes it tasty for the phagocytes. So the phagocytes are going to come in and more likely to grab it and eat it. It's intensifying inflammation, which means more antibodies, more leukocytes are being able to come out and come into the region there. So that's all. And then the last thing, obviously, we saw in our blood typing lab, agglutination, right? Antibodies end up clumping them all together and grabbing them together. So that's something you saw kind of more viscerally. You didn't actually see, you got to see pictures of it. But you kind of, you saw, quote unquote, saw. All right, so what we're going to do now is finish up our discussion of the immune system by looking at the other branch of specific immunity, which is cell-mediated, which is still it still shares all of these characteristics. It's antigen specific, it has memory, it's systemic. It's also going to be initiated by an antigen presenting cell, helping create clone armies, but it's a very different kind of clone army. Instead of clone armies of plasma cells making antibodies, we're going to actually have clone armies of killer T cells going out. But before we can actually talk about that, we have to introduce one other concept, which is MHC, or Major Histocompatibility Complex. So that's what we're going to do first as our lead into cell-mediated. So,
Professor?
Uh-huh.
Are T helper cells and T cells the same thing?
T cells are the more broad category, and there's many subtypes of T cells. So T helper cells are a subtype. Killer T cells are another subtype. There are also suppressor T cells. There's lots of different kinds of T cells. So cool. So T lymphocytes mean they just mature in the thymus gland. And we're going to talk specifically right now about two of the important ones, and there's actually other ones as well that we're not even going to talk about.
Got it.
MHC. So this is going to be major histocompatibility complex. So histo is just a prefix that means tissue, your tissues, your cells. So MHC is it's a membrane protein. It's a membrane protein, and it plays a couple of different roles. So let's talk about the two roles it plays. One is it is kind of your cell ID marker. You have a particular MHC. It's just a membrane protein on all of your cells. On, I'll say almost all your cells. It's not on red blood cells, but in general, almost all your cells have this marker. It has a very specific look, ID, and it's what your immune system is going to use to decide whether or not this cell is actually part of you or whether it's something that shouldn't be there. This is why transplants usually get rejected. Like, I might want to give you my kidney to save your life, and it might be it probably won't actually help you at all, because your immune system will look at look at the cells and say, those MHC proteins don't look right. They look like somebody else's, and let's kill it. If you're gonna get a transplant from somebody, you've got to find somebody who's genetically close enough to you where the MHC is really close, so it's not going to trigger such a strong response. And then you're still going to be on immunosuppressant drugs for the rest of your life, so you don't take out the donated organ. So this MHC protein is basically an ID marker that tells your immune system that this cell belongs to you. So that is one thing that MHC does, is just kind of mark it as you. The other thing that MHC does is it actually helps present antigens. So there's two kinds of MHC. So MHC1 and MHC2. This is the kind of normal one. MHC1 is the one that is on most cells. MHC2, this is specialized. This is on antigen presenting cells. So we'll come back to this one in a second. Let's start with MHC1, which is kind of your classic MHC. All right, so let's say here's my cell. Here's my MHC protein, MHC1. So this is just a normal body cell. So when the MHC protein is getting assembled, right? It's a protein. The cell has a DNA code on what amino acids to assemble in what order, and then it goes through all these different little vesicles and is assembled and folded and blah, blah, blah. As it's assembled, one of the things that gets integrated into this molecule is some random peptide fragment. So there's like little peptide fragments. Remember, peptide is just, like 7 to 8 amino acids. It's just a little chain of amino acids. So some little peptide fragment gets integrated and anchored on this thing. So this is some random, endogenous peptide. Endogenous meaning it was something that was just floating in the cell. There's little snippets of amino acids just kind of floating, and this little random endogenous peptide got integrated and is kind of in this little slot and is actually being presented to the outside world here. And why this is important is it's going to give your immune system cells a chance to get a view of what's going on inside the cell. Right, viruses, think about viruses are inside the cell, taking over the cell. And so even if you have, we're going to talk about like our killer T cells, kind of coming along, and, he's got like the little, in one hand, he's got his dagger to take out the offending, thing that it needs to get rid of. And in the other hand, he's specific immunity. So in the other hand, he's got like the specific shape, like he's looking for this particular thing. But, let's say that virus is multiplying inside the cell. He doesn't see it. He's looking. He can't see that the virus is inside the cell because he's looking at the outside of the cell. So this is supposed to be like my killer T cell here. But because these MHC proteins are getting assembled with little random pieces of whatever is floating around, if this cell is filled with viruses that are multiplying, there's going to be MHCs getting assembled, and the little random thing that it is displaying to the outside world is, oh my God, it's that. So now all of a sudden, this killer T cell is like, doop, doop, doop. It's like, bingo, wait a second. That is exactly what I'm looking for. So this is a way for your cell-mediated immunity to actually look and have a sense of what's actually going on inside the cell, right? There's no little porthole or no little window. This is kind of like the little window where the cell-mediated immunity is going to be able to actually realize this cell is riddled with virus. It has to be eliminated before it lyses and releases more virus to the rest of the body. So MHC1, part of what it does is a cell ID marker, but another role that it plays is it gives your immune system cells a chance to get a little glimpse into what kind of stuff is inside the cell. And if it happens to be an antigen that you're targeting for destruction, then you realize oh my God, this cell is, it would be if I, here's a little piece of liver. No, that looks normal. Oh, here's a little bit of heart. That looks normal. That should all be in me. But oh, here is okay, what do they call it? An exomorph, one of those aliens from that movie with Sigourney Weaver. It's that obviously shouldn't be in there. Something's really wrong, right? So here's a little sample of what's inside me. If it doesn't look right, if it looks like this kind of horrible parasite that we actually know is bad, that's kind of what this is about. It's giving you a little sense of, is this like the alien ready to burst out of your chest? So MHC1 displays a little fragment of some endogenous peptide. And again, it's going to play this role to allow the cell-mediated immunity, particularly these killer T cells, to see, is this virus that it's looking for actually present within the cell? The other one, MHC2, displays fragment of exogenous ligand. So this is the one that's on the antigen presenting cells. So we've already introduced this without calling it what it is. So basically I had my, my antigen presenting cell, maybe it's typically a big macrophage or something. And here was my little pathogen or whatever. And this macrophage is going to come and eat it. It goes raw. So it eats this thing, kind of dismantles it. But in that process, it's making MHC that is going to present a fragment of an exogenous, not ligand, what am I talking about? Exogenous peptide. So basically takes a little fragment of this thing it just ate. And now in its little MHC, so here's its MHC2, it's going to have some little piece of this thing that it ate. So that's when I say exogenous peptide, exogenous from the outside. It's some little peptide that it actually got from outside itself. It's not representing something happening in the cell. It's actually a little bit of something that it ate. So this is going to be the dude that's going to go and activate the rest of your specific immunity, right? It's going to say, here's a little peptide fragment of this thing I ate. Use this as the antigen that you are going to use as the target for your specific immune response here. right? So MHC2 is only found on these like antigen presenting cells rather than a more generic thing like MHC1. So any questions about MHC? You know, one of the things one of the fascinating, I'll just say this briefly just because it's so kind of interesting if you haven't heard about it. You know, if you use MHC as a kind of measure of, you know, kind of genetic closeness or, you know, being more genetically similar or more genetically far away from another person, right? Because if you have, if your MHCs are really close, I can actually receive or give, like share organs even. When MHC is really different, the two bodies are really like, I don't recognize you, I don't recognize you. So it's more kind of more genetically diverse versus more genetically similar. People have done these experiments looking at human pheromones. You know, so it's worth mentioning just for a moment, even though we don't have much time. You know, when we've talked about like smell, you know, and this is supposed to be your nose. And we talked about the chemoreceptors up in your olfactory, you know, olfactory epithelium, chemoreception being smell. There is also airborne chemoreception. You know, so there's molecules in the air. They are bound and recognized by these chemoreceptors in your nose, and that's olfaction. So this should be familiar from what we were talking about before. It's always been known that in other animals that there's a similar system that is not about smell, but instead it's almost like airborne hormones. In those what we call pheromones. And so they're molecules from the air that you come in and they bind to chemoreceptors, but they're not sensed as olfaction. Instead, they initiate other kinds of physiological responses in your body. And for a long time, it was like, oh, all these other animals use pheromones and are influenced by kind of these molecules given off by other members of their species, but not humans. We're so advanced. But now it's more and more clear that there is some of this stuff likely going on with humans as well. And again, it's something where there are molecules floating in the air that somebody else is giving off that you bring into your system, are detected by chemoreceptors, but you aren't experiencing a smell, but they're affecting you in some way. And so one of the really fascinating studies that, you know, is suggestive, I wouldn't say this is true, but is very, it's very intriguing, was they had people like wear, I think they wore t-shirts and then would put the, you know, the t-shirt after they'd worn it is like in a kind of a box that somebody could smell. So you can smell like this, you know, this shirt that's kind of absorbed these whatever molecules a person's giving off. And you had to kind of decide whether or not, is this more pleasant, less pleasant? And what they found, the ones that people liked, and it wasn't like, oh, like, wow, that's really sexy smelling, or ooh, that's gross. It was more like, yeah, I don't like that, or yeah, no, that's pleasant. Or sometimes the pleasant, when they asked them, what do you mean by pleasant? It's like walking into the, like kind of nice, it was more like comfortable, like it wasn't like about pheromones being like, oh, these are turning me on. It was just more, I like it. It's something I like. And what they found was there was this total correlation between whether or not you liked that smell. And not even smell, it wasn't a smell, it was like an experience, whether the experience was positive or negative. And it was correlated with how genetically similar or different the MHC of the person was to yours. So it seems that there is this thing going on subconsciously where when you are near somebody and your MHC is really close, this is your MHC and their MHC, if these are like really genetically similar, it's like, nah, it's like, I'm not really, this isn't, or if they're too far away. But if there's like this kind of like sweet spot for like, you know, genetic diversity is kind of the thinking here. It's like, then there's just something like, I'm just feeling this. There's something just nice about this, that there's actually something going on that makes you more likely to be open to like, this just feels nice. That's happening subconsciously by molecules coming up your nose from somebody else because there's a nice genetic distance. So like, you know, again, the thinking is if you're going to make babies together, there's going to be kind of good genetic diversity. So it's kind of hilarious. So anyway, I just wanted to throw that in. Whenever I think about MHC, I always think about this study. And there's been a few studies that have looked at similar things. And again, it's still something people are trying to understand more, but there's enough evidence that there's likely something going on with this. So anyway, let's go back to our regularly scheduled program. Let's talk about T cells and cell-mediated immunity. So this is the other side of the specific immunity. And the first parts are going to be similar. There's going to be T cells with particular receptors, finding some antigen presenting cell that's presenting its little antigen, and they've got this co-stimulation, and you're going to basically have clonal selection again. So the first part is going to be similar to what we saw for the B cells. The difference is the population of cells that get created. Instead of making plasma cells to make antibodies, we're going to be making one kind are called, they have different names, cytotoxic T cells. These are also called killer T cells. These are also called CD8 cells for like cell differentiation factor 8. So cytotoxics, and these are basically, these are like the natural killers, except they look for specific antigen. So they basically, this is the dude I was drawing over in this picture here. This little killer T cell, he is looking for a particular antigen, and if he sees it, most likely displayed on the MHC of some cell filled with viruses, he is going to insert these perforins, put holes in, and kind of take out this virus-infested cell before it can cause damage to the rest of the body. So here, the cell itself is the agent of destruction. Rather than antibodies having this more indirect process to take out the target, the killer T cell puts in these perforins, these proteins into the membrane of the target that fill it full of holes. So killer T cells are part of the clonal selection in cell-mediated immunity. The other ones are called helper T cells. Also called CD4 cells. These are necessary for activation of both cell-mediated and antibody-mediated immunity. I'm going to say of. So I talked about that big complicated co-stimulation kind of thing going on. You need helper T cells as part of that for actually both aspects. You know, and the reality is the activation and control of the immune system is something we're continually understanding more and more about. Every week in like science, there's never fails, there's another article about some deeper understanding or changing our understanding of how this whole thing works. Like everything I'm telling you so far is the basic understanding of like really broad aspects of the immune system. But when you get down into the specifics or the more subtle details, it gets so complicated. It's a rabbit hole. The whole thing with, you know, even like allergies or like, you know, if you start seeing the same thing again, sometimes you get more and more sensitive to it, or sometimes you get habituated and you get less sensitive to it. And when do you go one direction or when do you go the other direction? There's so much stuff that's complicated and not well understood about the immune system. Part of why it's important to know about helper T cells and knowing that helper T cells are necessary for both sides of the immune system is these are the dudes that get attacked by another nasty virus, HIV, the human immunodeficiency virus. You know, if, you know, HIV, you know, attacks, there's two parts that make it particularly nasty. So it attacks helper T cells. So helper T cells, which are critical for the activation of your immune system. And it's a retrovirus. It's like an RNA virus. It mutates super fast. Like super fast. So basically, it comes into your body and it's a virus. So your immune system has a reaction against it, you know, and it's going to make memory cells against it. But before you can actually remove it totally from your body, it has mutated enough that it looks different enough that your immune system response against it no longer recognizes it. You're looking for, you know, it's like, you know, it came in, you know, rawr. You know, and it's starting to like destroy your T cells, and you do have an immune response, but then it's like, you know, all of a sudden it's like, oh wait, I don't recognize, that's a virus got a mustache. I don't know what that one is. And all of a sudden it's starting to just continue to take out your immune system cells. And then you can start creating a, you know, immune system response against that. But then, well, I don't recognize that one with a hat, you know, and then, so it just keeps going. And while it keeps sliding under the radar, it's then dismantling your immune system to where you can't fight it or anything. And you start getting sick from the normal little like things that normally your immune system would easily be able to take out. So you start getting things that normally aren't issues at all. You know, you start going into AIDS, you know, the acquired immunodeficiency syndrome, where you basically can't fight off anything. Like the normal yeast that would normally just land in your mouth and you'd kill off now is just coating your whole inside of your mouth in this white film and thing, you know, so it's like, that's why this is like, you know, it's not as it used to be, like a total death sentence. And now they have a lot of drugs that can kind of inhibit its activity so it doesn't progress this way. But that's, you know, when you think about helper T cells, that's what this is attacking. And that's what causes this, is taking out the helper T cells, takes out, you know, basically dismantles your ability to have specific immunity. You know, you'll end up with AIDS there. While we're talking about like pathology, we can talk about, you know, transplant rejections. Again, this is because MHC on another person's cell is different from your own cells, and you're going to not recognize it as your own cell, and your immune system will attack it. So like I said, if you want to have a successful transplant, you've got to get it from somebody who's genetically similar enough that their MHC is pretty close, so you'll have less of an intense immune response against it. And even then, you'll be on immunosuppressant drugs the rest of your life, so you don't kill off your donated organ. Autoimmune diseases. You know, these are when you start creating a specific immune response against an antigen that's actually a normal part of your body, so this is horrible, right? So this is like, you know, MS, multiple sclerosis, you're attacking myelin sheaths on your neurons in your central nervous system, and all of a sudden your neurons aren't going to be able to send the messages where they're going because the signals die out along the axons. Rheumatoid arthritis. This is when you attack the synovial membrane in your joints. So all of your joints, normally the synovial membrane is making the nice slippery fluid, so keeping your joints all slippery and moving nice. But now the synovial membrane is getting attacked, it's getting all inflamed, your joints are painful, they're starting to seize up. And this is like really nasty. This happens to people really young. You know, all of you, by the time you're like 80, are going to have creaky joints with osteoarthritis. That's just part of getting older. Your cartilage wears off and your joints hurt. But this is like, you know, 20-year-old person who can barely move their body. I've had students who are like, again, in their 20s, had already been through multiple surgeries and on super intense steroids and stuff, just trying to be able to live a normal life. So this is really, really nasty. Lupus. I think it's official name, Lupus erythema, something or other. This is kind of a more generic. You're just kind of attacking all of your tissues. So like your organs, just your general organs start having inflammation as your immune system starts attacking its own organs. So that's kind of this idea of autoimmune diseases, is when your immune, you know, that really tight control of only activating it if it really should get activated. If something goes wrong and you start attacking part of your own body, it can lead to all sorts of horrible conditions. You know, and they're tricky to treat, because if you suppress the immune system so you don't attack your own body, then you're also suppressing your ability to defend yourself against actual dangerous things as well. Or even transplant, like one of my friends who had a liver transplant, you know, had to be so paranoid about just being in the room with somebody with, you know, that could be, you know, would be sick or something, because, you know, the drugs kept him alive so he could have a good liver, but it also meant that now it was kind of more like the boy in the bubble. Had to be really terrified of like a normal kind of thing that would just make somebody sick and they'd get better, or maybe wouldn't make them sick at all, could kill them. So anyway, so immune system, there's way more we could talk about, but it's pretty cool. And especially now, I'm sure you're going to be learning much more about it on your own as we continue figuring out how to address the COVID-19 deal. And as they're trying to figure out, you know, how to come up with ways of dealing with the epidemic. So any comments, questions, or anything about all of this stuff? All right, so that is, we're going to leave that behind. So digestive system now, which we have introduced. So we've already talked about how it's about breakdown and absorption. Right, we're just, food is basically other creatures with biomolecules assembled into a cow or a broccoli plant or something. We need to disassemble those molecules into basically the monosaccharides and amino acids and such. Then we have to absorb them. And then we can do two things. We can either like reassemble them into our own form, or we can break them apart for energy. It's not a bad idea just to kind of do a quick kind of rundown of what we, you know, in nutrition, you do this in more detail. But when we talk about, you know, what are nutrients even? And what are the things we actually absorb and how can we use them? You know, obviously, from our last class, there's carbohydrates, there's proteins, there's lipids, you know, fats. These are different from all the rest in that these can all be broken down for energy. The other ones we're going to talk about aren't broken down for, they're necessary, you know, you need them, but you don't have the option to disassemble them and release energy from the bonds. You know, and you don't always. Some of the amino acids you're going to use to just assemble into your own proteins. Some of the fats you're going to use to make your steroids and cell membranes and stuff. But you can also break them apart and feed them into all that cellular metabolism to, you know, make ATP and stuff. So these are nutrients that you can get energy from if you need to. Other nutrients we talk about minerals. These are just elements. This is like, you know, calcium, phosphorus, magnesium, whatever, right? So mineral, when we talk about, you know, lots of minerals, minerals are just elements. Anything on the periodic table is officially a mineral. Sodium. Vitamins. So vitamins, these are essential organic molecules. So essential, let's talk about essential. Essential nutrients. So when we use the word essential in the terms of nutrition, it has a very specific meaning. Essential means must be acquired through the diet. So for instance, like there are no essential carbohydrates. If you don't have glucose, you run gluconeogenesis. Any carbohydrate you need, you can make using the other stuff you've got, using your metabolic pathways. So there's no essential carbohydrates that you need to get through your diet. There are essential amino acids. There's somewhere, depending on what you read, 8 to 9 essential amino acids. You know, there's 20 in all that are used to make the proteins in your body. And of those, a little less than half are things that you have to acquire through diet. This is where you get into the idea of complete and incomplete proteins when we talk about different kinds of food. If I have a food that I call a complete protein, you know, food has all essential amino acids. You know, in general, if you are eating something that has a body plan similar to you, it's going to be a complete protein. If you're eating another animal, if you're eating a cow or a chicken or something, another thing that's a lot like you, it's made of the same stuff as you, and it's going to have all the different amino acids you need to use for your own body. If you're eating an organism that is more alien, that like a plant is a really different kind of organism from an animal. It doesn't have all the essential amino acids necessarily. So we have the idea of an incomplete protein. You know, does not have all essential amino acids. Right, so like for instance, wheat. Wheat has amino acids, but it does not have all the essential amino acids that you need. So if all you had was like wheat bread, you might have enough energy to run everything you need in your body. You might be full, but eventually you'd have this horrible form of malnutrition called kwashiorkor because you're missing some of these essential amino acids that you need to make the proteins, and your plasma proteins aren't being made properly. All the fluid is leaking out of your vessels into your tissues. It's all gross. So, you know, so you can have horrible malnutrition and eat all the bread in the world because you don't have all the essential amino acids, because wheat is an incomplete protein. Is it possible to be a vegetarian and get all your essential amino acids? Obviously, there's a lot of vegetarians out there that aren't like dying. So how does that work? How can you get all your essential amino acids even though most plants are incomplete proteins?
You have to pair things like rice and beans.
Yeah, exactly. So they call them like complementary proteins. So one, like the rice doesn't have everything you need. The beans don't have everything you need, but the things they're missing are different and they complement each other, and together they give you everything you need. So the basic idea is you have to be a little more intentional about your diet if you're a vegetarian, right? If you are going to eat meat, you can just be kind of brainless. It's like, oh, if I just shove this chicken in my face, I'm getting all my amino acids. But if you're eating plants, you need to be a little more conscientious to make sure that you're getting a good mix to get all of the amino acids that you need. So no essential carbs. There are essential amino acids. There are 2 essential lipids. This alpha-linolenic fatty acids. These are like those omega, I forget what they call them. But yeah, there's a couple of essential amino acids. And then this takes us back to vitamins. A long way back to vitamins. Vitamins are essential organic molecules. So essential meaning you need to get them from your diet. Organic molecules meaning they're not like these elements. They're carbon-containing molecules. And these are usually used, as like coenzymes. So these are used to help run the metabolic pathways of your body. So these are things that work together with your enzymes to run the metabolic pathways. So, like think about niacin and riboflavin, the B vitamins. Those are just the precursors you need to make NAD and FAD to run cellular respiration, right? That's what niacin and riboflavin are. Those are just the things you need to make NADH, right? Niacin is to make NADH, which we all know is just the high energy electron carrier to run cellular respiration. So that's what I'm talking about. These are molecules you need to run your metabolic pathways along with your enzymes, and they must be acquired through the diet, which is why we call them essential. So that's what vitamins and minerals are. And then often people talk about, water is also considered an important nutrient. If we're going to do completeness, water is like a nutrient. So that's, and for our class, we're going to be focusing on, like right now, most of the liberating and absorption of these carbs, proteins, lipids. We'll talk a little bit about this as well, though. So let's dig in. All right, like I mentioned last time, the GI tract is basically a tube running through you. It starts at your oral cavity. There's the esophagus. You go down into the stomach. You get the small intestine, the large intestine, the rectum, and then the anus. And then you come out the other side. So this tube has a couple of different names. Can be called the gastrointestinal tract. Usually called just abbreviated the GI tract. It's also known as the alimentary canal. And the basic idea is the food is never actually in your body proper, right? The food comes in, comes down, it's getting kind of broken down. Whatever's ultimately not being used is coming out as your feces here. But it's never in your body. It's basically a tunnel running through you. So we're going to talk about what's going on in this tunnel that's actually breaking down the food. Then we're also going to talk about the absorption. The absorption is going to be pulling the nutrients across the wall and so they can get absorbed into your bloodstream. So we're going to talk about the breakdown is occurring within the tube, and then the absorption is going to be occurring across the wall of the tube to get absorbed into the bloodstream. We're also going to talk, so we have the GI tract, and there's also what we call accessory organs, accessory digestive organs. Going to be like your teeth, the salivary glands, your liver, your pancreas. So we'll get to meet them as well. And so these are the things that are helping with the breakdown, either by crushing or by providing essential secretions that are going to help with the breakdown. So let's come back. So some of this will just be kind of retreading the stuff we introduced in the lab last time. This idea of breakdown is going to start in the mouth. We need to start clearing. What? Okay. So your teeth are going. You have like your salivary glands pouring saliva in. This would be like my the parotid salivary gland is the one that is kind of in your cheek. You've got submandibular salivary glands pouring in, sublingual salivary. You've got a variety. Don't worry about the different salivary glands. Just know that they exist. Then we have our pharynx, the throat. Here's the esophagus going down towards the stomach. So this is kind of a good introduction to the two kinds of breakdown that are occurring in the digestive system. When we talk about breakdown of the food, the first thing that happens is you start chewing it. So that's called mechanical breakdown. Mechanical breakdown, this is when the food is still kind of more intact and you're not going to be able to disassemble a hunk of bread into like individual like glucose molecules until you start kind of pulverizing it and increasing the surface area that enzymes can actually work at. So mechanical breakdown is going to be like chewing. Actually, even once the food ends up in your stomach, it's still pretty intact. Like if you've ever like, you know, if you've eaten a meal and barfed really soon after, you recognize what you've eaten, right? It's not like the food drops into your stomach and it's all of a sudden liquefied goo. It's like you barf and it's like, oh, there's my chewed up spaghetti or whatever. So the stomach, you know, kind of kneading. We're going to talk about the stomach. The stomach has these muscles that are just going smush, 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 kind of taking your food and really mushing it. So mechanical breakdown, there's no actual turning it into, it's not breaking chemical bonds yet in the way of liberating individual biomolecules. Instead, it's about just this more bigger picture kind of mushing it up. At the same time, we saw the salivary glands actually are starting with the second part, which is the chemical breakdown. You know, this is using the enzymes. This is the part that's actually going to allow us to end up liberating like individual monosaccharides and amino acids and things like that. And it's going to happen in stages as well. You know, the amylase from the saliva, it does not liberate individual glucoses. It starts by kind of breaking the starches into smaller chains. It's not till you get into the small intestine that we're going to have the enzymes that liberate individual monosaccharides before we absorb them. So this is useful to know, though, the distinction between mechanical breakdown, which is more in the beginning of the process, And then as we get further down in the system, it's going to be more and more just pure chemical breakdown, where you are just kind of mixing the food with different enzymes and just keeping it going until everything breaks apart. You know, the very first part of this whole process, we also talked about the salivary glands having amylase. That is part of the chemical breakdown. We also talked about it having mucus to help bind the food into a little gooey ball so you can swallow it. Swallowing is got an official name called deglutition. Deglutition is the official word for swallowing. And it starts out being voluntary. You kind of push the food back to the back of your throat, and you have these muscles, pharyngeal constrictors, that can squeeze and push it down. How do we make sure that the food doesn't go down in your windpipe, into your trachea, and choke you?
The epiglottis.
Yeah, you've got the epiglottis there. It's going to make sure that the food goes down the right tube. And now what we're going to do is look at this tube in more detail and see what's going to happen now as this food enters into that crazy, you know, tube running through you. So we're going to look at the GI tract in a little more detail. So let's do that. So this is like a cross section of the tube. You know, I'm not going to go into the crazy detail that we would like in anatomy, but just to give you some basics here. You know, the layer that is lining, so the very most inner part of the tube is called the lumen. The epithelium that lines the lumen, we just call this the mucosa. You know, on the other side of this epithelium is where you're going to find the capillaries and stuff, where you're going to actually be able to absorb your nutrients. So the nutrients are going to have to, it's going to be transepithelial transport, right? It's going to go back to our first week of class. When we look at how do we bring sugar from the lumen as you break apart the food and you absorb it into the capillary, that was on your first exam. Remember that, with all of the, in fact, that was the actual example. It was the intestinal lumen was where the sugar was, and the little transporters were getting you across the simple columnar epithelium of the intestinal mucosa so you could absorb the nutrients into your bloodstream. So, you know, this absorption part, at least for sugars and proteins, you actually know already. Just it's transepithelial transport like you've already seen. The absorption of fats is going to be trickier because fats are hydrophobic. So those don't just float in the water, and you can't just do the same little trick to absorb them. So we're going to see a more roundabout way to absorb fats into your bloodstream. They're actually not going to go straight into your bloodstream at all. They're going to end up getting absorbed into lymphatics and then ultimately dumped back into your bloodstream. And then we also, again, we talked about when nutrients are absorbed into the bloodstream, do they just go directly into circulation?
Go into the hepatic portal system?
Yeah, exactly. They actually first get absorbed into the capillaries of the GI tract, but then go into the secondary capillary bed in the liver. And so things all get processed there and detoxified, and you can store nutrients or convert nutrients or do whatever. But that means if you're absorbing nutrients through your, or absorbing anything through your GI tract, it's going through the liver first, which has a lot of implications for kind of the efficacy of a drug. Like if for a drug to be effective as an oral drug, it means it has to be able to survive going through all those enzymes in the liver that are designed to break things apart and detoxify things. So there's a lot of drugs that you can't administer orally. You know, that's why there's things that you have to inject, or you have to do sublingually, or, you know, rectal suppository or something, get it directly into the bloodstream without having to go through the, you know, hepatic portal system and getting dismantled in the liver. So anything that you take orally that gets absorbed into the, you know, across the mucosa of the GI tract is going to have to end up in the liver. So that's why there's a lot, yeah, again, things that are active orally, there's things that are not active orally, but, you know, are very active if you snort them or if you inject them or whatever else. You know, below the mucosa is the submucosa, is just kind of connective tissue under there. And then under that is the Muscularis Externa. We need to talk about the Muscularis Externa a bit. So there is smooth muscle. There's two layers. There's one layer of smooth muscle that is circular. So this is my circular layer. And then there's a longitudinal layer of smooth muscle where the fibers are going the long ways along. So I use my little text here. Inner circular layer, the outer longitudinal layer. What that means is you have the ability to contract this inner layer. It's going to squeeze the tube. If you contract these longitudinal muscles, it will kind of pooch out the tube. So you can either squeeze the tube or kind of pooch the tube. There are two specific types of contractions of the GI tract that we talk about. We should just add them in here right now. So the two classic ways of contracting these muscles, we have peristalsis, you know, which is for propulsion, for moving things along. We have segmentation, which is for mixing. The basic idea is, let's say, you know, the esophagus, for instance, is pure peristalsis. You've got your food that you've just pushed down to the back of your throat through deglutition, and now it's at the top of the esophagus, and now the automatic reflex takes over. Basically, the circular muscle above it is constricting, squeezes in. You know, you pooch out the bottom. And so as you squeeze on the top and relax the bottom, it gets pushed down a little bit. So time-lapse photography here, it's a little further down. And when it's here, now we can squeeze the top of it now. And so basically you've got this wave of contraction above and relaxation below that keeps pushing it along one direction. You know, that's peristalsis. When you swallow your food, that's how the food gets from the back of your throat to your stomach, right? You can be lying on a couch and somebody can be feeding you grapes as you just lie horizontal. And as you chew up your grapes and swallow them, they still get to your stomach. They don't need gravity. They just get squished along by peristalsis with the smooth muscle in the wall of your stomach, of your esophagus. You could even be lying, standing on your head and swallow, and the food would make it up to your stomach. It also means it takes, this is a lab we're not going to do. We actually, one of the labs that we do sometimes is use a stethoscope and like listen to when you actually hear the food drop into the stomach after you swallow it. So if you swallow, it takes about like 5 to 7 seconds before the food actually drops into your stomach. Like, it's like, whoop, 1 and 2 and 3 and 4 and 5 and 6, swoosh, and all of a sudden you hear it drop down into your stomach. So it takes a little while. And if, I'm sure you've experienced this either swallowing a piece of food or a pill or something where it's like, ugh, it's stuck. Something didn't happen right. It's like, I feel this is uncomfortable. So if that happens, if the food gets stuck and the wave continues down, so sometimes like the wave is continuing down, but the food got stuck, in which case the system will just initiate a secondary wave that will catch it and continue, moving it down. So peristalsis, propulsion, and then segmentation for mixing. So mixing, once you're down in the lower parts of the digestive tract, a lot of it is going to be mixing the food with different digestive enzymes and just smushing it around, allowing the enzymes to do their work. So in that case, it's not about moving things along. It's just about more like a washing machine, keeping things mixed. So kind of the classic way to draw it. Let's say I'm in the intestine here. For clarity, I'll have green food and blue food. And the basic idea now is instead of squishing to move things in one direction, you kind of squish where it's bulked out, you pooch where it's squished. And so now in our time-lapse photography here, we've just kind of moved the food into other sections, and we squish again, and the food's just getting totally squished up in here. So it's just kind of squish, 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 back and forth, back and forth, back and forth, squish, squish, squish. And so the food isn't going anywhere, it's just getting all mixed up. So that's segmentation. And obviously, it's usually kind of a mix. The esophagus really is pure peristalsis. You're just getting the food from your throat to your stomach. But then the rest of the time there's mixing, but then moving a little along as it's getting processed, because obviously it's got to make it to your anus by the end. So there's got to be propulsion to get to the very end. But then a lot of mixing along the way too, so the breakdown has a chance to occur. So peristalsis for propulsion, segmentation for mixing. The other thing I'll mention, kind of big picture about the GI tract before we take our break, is in addition to the mucosa and submucosa and muscularis externa, there are also nerve plexuses, networks of neurons within the wall of the GI tract. There's neurons in the submucosa, the submucosal plexus of Meissner. There are neurons in between the mucosa and the muscularis, the myenteric plexus of Auerbach. So let me use the typewriter tool because it'll make it easier to type it. So we call this the enteric nervous system. There's the myenteric plexus of Auerbach, the submucosal plexus Meissner. Sometimes they call this the primitive, I'm going to put it in quotes, The primitive gut brain. So the basic idea here is a lot of the processes of the digestive system don't actually need your brain. Remember, reflexes were, let me just go back here. Reflex is stimulus, integration, response. So there are a lot of reflexes that are part of your digestive processes that are integrated completely within this enteric nervous system, integrated within the walls of the digestive tract. They don't use your brain. We call them short reflexes. If they're integrated within this enteric nervous system. You know, what are the stimuli? What kind of stimuli do you think are part of this enteric nervous system? What kind of receptors? What is it going to be monitoring?
Stretch receptors?
Totally. So the stimuli are going to be stretch receptors. What's another one?
Chemoreceptors.
Totally, chemoreceptors. So it's very similar to what we did. Think about the carbohydrate lab, where if we want to do like kind of watch what was happening with digestion, we want to look at, you know, is the substrate there? Is it not there? Chemoreceptors for the substrates of digestion. So you know, what kind of enzyme do I even need to break stuff down right now? You know, chemoreceptors for the products of digestion. So you know, are we done yet? Have we broken it down? Has the substrate disappeared and the products appeared? Right, so you're gonna need this kind of information if you're gonna control the digestive processes. You need chemoreceptors for both the substrates and the products of digestion. You know, so you have receptors for this stuff. You know, what kind of responses can you have? What are the ways, what kind of mode of action can this enteric nervous system actually reach out with? It's the same kind of motor stuff that we see in the other kind of nervous system as well.
Produce an action potential.
No, that's how you would send messages. But what would be the actual physical result of that? What are you activating? You're sending an action potential down a motor neuron to do what?
Moving the muscles.
Exactly. You know, squish the muscles. You know, do segmentation or peristalsis, for instance. So one of the responses is just muscles. You know, there's, you know, squish things, mix things, move things along. What's another kind of response you can have? I mean, what would you need? Let's say the chemoreceptors just found a bunch of starch in your intestine. What are you going to need?
Enzymes to break it down.
Yeah, so secretion, right? You can tell glands like, please secrete, I need stuff. So, I mean, it's the same thing when we talked about, you know, motor response, you know, when we talked about the more generic nervous system as a whole, you know, kind of motor response. Usually it's either some kind of muscle or some kind of glandular activity. So it's the same thing with this enteric nervous system. Sometimes you're secreting things directly, like telling glands we need some juice. Sometimes you're secreting hormones. So So this idea of short reflexes, and at least for me, I know it was pretty weird and trippy the first time I realized that, your GI tract at some level has this level of autonomy, that it's doing its own thing, that doesn't need the brain to take care of business. That being said, there are also long reflexes. So I'll leave it by mentioning the fact that there are also long reflexes. So you have short reflexes that are integrated totally within the enteric nervous system. But then there are also long reflexes. We're going to talk about this a little later. For instance, just like thinking about yummy food, you're going to have signals coming down your vagus nerve, right? Your parasympathetic activation, starting to activate your digestive system, getting it ready to digest stuff. So it's not like the brain can't be involved or is never involved. It's just that there is a lot of cool stuff going on where it doesn't have to be involved. So any questions about that? Okay, so let's take a break. We'll come back at 10 o'clock, and at 10 o'clock we will get ready to begin our quiz, which will be pretty quick. And then I think, for the rest of today, we need to do two things. I do want to kind of finish up the digestive system and review for the lab exam, which is going to be coming up. So let's take a break, and I'll see you all back at the ranch at 10. So last we left our food bolus, we were talking about the mechanical digestion of chewing and beginning the enzymatic digestion in the mouth with the salivary amylase breaking the starch down. And then deglutition, we pushed the bolus to the back of our throat. Then we got peristalsis, carrying it down the esophagus. And then we drop into the stomach. Just to give you a sense of the big picture, let's do the big picture, and then we're going to get back into each of the different sections. Maybe I can do a different color. From the stomach, you're going to head into the small intestine. So stomach. The stomach is going to play a number of roles. A big part of what the stomach does is just storage. It allows you to eat a bunch of food and slowly send it down to the next reaches of the digestive tract. If you didn't have this, it can hold up to a gallon. So you can eat a bunch of stuff and then sit and watch me on Zoom while your digestive system is working on it and squirted it out a few mils into the rest of the GI tract here. It's also going to be involved in protein breakdown. We'll talk about that in a little more detail. And a little more. We'll talk about a little more. Little absorption. It's not much about absorbing. The only absorption that happens in the stomach is maybe water, some drugs, things like alcohol. If you were drinking margaritas like last night, and you are buzzed pretty quick, it's because the alcohol crosses the stomach lining and it gets absorbed into your bloodstream right away. So you don't have to wait for the tequila to make it down to the lower reaches of your digestive tract before you absorb it and start feeling it. So alcohol, aspirin, things like that can get absorbed across the wall of the stomach. Water gets absorbed across the wall of the stomach. But that's about it. In general, this is not a place for absorbing. This is a place for storing food that you've just eaten a big meal. It's a place we'll see breakdown of protein is happening. The small intestine, on the other hand, this is main site of both breakdown and absorption. So, in fact, it continues breaking down proteins. It continues breaking down the carbohydrates. We'll look at it in a little more detail. But basically, small intestine is the main place where the lion's share of both breakdown and digestion are occurring. And then the final part is going to be the colon. Also called the large intestine. So a big part of what's going on in the colon, all along this digestive tract, you've been pouring all these digestive juices, enzymes and things to mix up with your food and break things down. And you've got this big slurpy goo, and you want to pull most of the water back into your body, before you get rid of the wastes. So this is a big part of large intestine is like water balance. Reabsorb H2O from all of this stuff. Sometimes they say, compact the feces. You don't want to pull too much water out, because then it turns into a rock and then you're constipated and you can't get it moving out. But if you don't pull enough water out, then you've got diarrhea and you are going to get dehydrated because you're losing too much water. So that's why I'm saying water balance. If you take too much water out, you actually are going to be in trouble because you're going to get constipated. Worst case, you can take so much out, it becomes like a rock, and then someone has to go in and physically chip it out. It's called like an impacted bowel. It's like kind of gross. All right, so we're going to look at each of these in turn. We're going to look at the stomach. We're going to look at the small intestine. We're going to look at the colon. And let's start with the stomach. And the stomach will also give us a chance to address some bigger picture issues around the digestive tract. So the stomach. This is like to the small intestine here. It's worth knowing some of the basic anatomical stuff. This is called the cardiac sphincter. Also called the gastroesophageal sphincter. That's basically controlling the connection between the esophagus, which is coming in, and the stomach. This is called the pyloric sphincter. Basically controlling access from the stomach to the small intestine. And this kind of big floppy part of the stomach up here is called the fundus. So when you're eating your food and it's kind of dropping in here, you know, it kind of lands here in this big kind of floppy fundus area. Like I mentioned, if you chew up a meal and you vomit pretty quick, it hasn't turned into goo yet. It still looks like just chewed up food. It looks like whatever you swallowed. So what has to happen once the food is in here, we start mixing it with different digestive juices and then start this kind of kneading process. There's smooth muscle. There's actually 3 layers of muscle in the stomach compared to the 2 in most of the rest of the digestive tract, where the food kind of gets squished back and forth, you know, up and down and up and down and up and down and up and down. And that's part of this mechanical digestion where you're smushing the food up and down while you're mixing it. We'll be talking about mixing it with acid, mixing it with pepsin that you've met in the lab. And as it turns from chewed up food into just really this white goo, the white goo we call chyme. By the time it's chyme, it doesn't look, you're not going to recognize the chewed up spaghetti anymore. It's just going to look like white goo. And as that is ready, it's really kind of processed enough, then it gets squirted out a few mils at a time down into the next stages of the digestive tract. Let's talk about the gastric pits and gastric glands. So here's my lumen of my stomach. Some epithelium, actually simple columnar epithelium. Then I've got, it's called the gastric pit. And then down at the base, there are all these glandular cells that makes these different secretions. And then they're going to be making stuff that's going to be going up into the lumen. So we should talk about what kind of cells do we find in a gastric gland and what kind of stuff is going up into the lumen. So let's do that. One kind of cells you got are called chief cells. And they make your old pal, pepsinogen. There are parietal cells. These are going to make hydrochloric acid. And, oops. And let's add in, there are also going to be enteroendocrine cells. So let's look at each of these in turn. Actually, let's start with parietal cells. Parietal cells, they make hydrochloric acid. Hydrochloric acid is playing a few roles in here. So the pH in the stomach, it gets down really low, pH around 2. It's really acidic. Remember, HCl is strong acid. It's going to kill bacteria. This is part of your, general defenses. You swallow stuff and it lands in acid. That's one of your body defenses. HCl, it's also going to help denature proteins. In order for the proteases to be more efficient, it helps to like unwind the proteins and make all the peptide bonds more accessible to getting hacked apart by the enzymes. Right? It's like if you're all kind of like this, it's hard to like get you, but if you unwind, then the enzymes come in and slit your neck or whatever. So that is part of what the HCl can do too, is kind of denature, unfold the proteins and make them more vulnerable to getting hacked apart by the proteases like the pepsin. The intrinsic factor, this is necessary to absorb vitamin B12. Which is used for lots of things, including making red blood cells. When people talk about like the stomach in terms of essential functions, this is the one thing that is an essential function that the stomach does that nothing else can do. In terms of, storage, we talked about how the stomach is good for storage. But if you don't have a stomach, then you just eat little bits at a time. In fact, people get surgeries to reduce the volume or staple their stomach if they actually want to lose weight, so they actually can't have a storage tank. So it's not like you need to have a storage tank. It's just really convenient. We talked about it doing protein breakdown, but we're going to see that your small intestine actually does plenty of protein breakdown. It's got trypsin coming in from the pancreas. So if you didn't have your stomach, you'd still break down protein just fine. The one thing that you would not be able to do is absorb B12 if you didn't make intrinsic factor. In fact, people who have these stomach reduction surgeries need to get additional, shots of this stuff to make sure they stay healthy. So intrinsic factor, the stomach makes it and you need it if you're going to absorb this vitamin into your body. Chief cells, they make pepsinogen. Have I talked in this class about pepsinogen versus pepsin? Yes or no?
The precursor?
It's a precursor. And, so one of the things we need to kind of talk about a little bit is this protease, the acid and the pepsin, it's designed to dissolve animals into amino acids, right? It's designed to take something a lot like your body plan, except it's a cow or a pig or something, and dissolve it. And how do you keep from just dissolving yourself from the inside? You've got all of these super corrosive chemicals, enzymes that are designed to dissolve flesh inside a bag made of flesh. So how is it that you don't just like dissolve yourself from the inside?
You have the mucus.
So there's going to be a number of things, and a big one is mucus. There's going to be a thick protective mucus layer along the inside of the wall here. And if I'm drawing, this is kind of so there's this protective mucus. That's going to be one way. So you have the kind of corrosive enzymes, but it doesn't actually get to the cells because there's this barrier of mucus in between. Another way that you keep from digesting yourself is we have the chief cells, which are sitting here in the gastric glands. they don't make pepsin, they make pepsinogen. Pepsinogen is not active. So pepsinogen goes up through the gastric pit. It's not going to harm your own cells because it's not active. It's not digesting protein yet. But when it hits the low pH in the lumen, then it activates. So in the low pH, basically pepsinogen, I gotta spell this right. Pepsinogen becomes pepsin. So that's another way you're protecting yourself from digesting yourself. The enzyme doesn't actually get converted into its active form until it's actually in the lumen in this specialized pH environment. For the other parts of the digestive tract, you have actually alkaline in the lumen, but it's the same basic difference. You have enzymes produced in an inactive form. They can get transported around and stuff without causing damage, and they don't actually activate until they're dumped into the lumen, into this different pH. So that's another way that you keep from digesting yourself, is make things in an inactive form that don't activate till the lumen. And then another thing is the cells don't have a very long life. If you are on the front line in the mucosa of your digestive tract, you're going to die pretty soon. You wear out. And so there's just a very high cell turnover. So there's mucus. There's this idea of inactive turning to active enzymes, and then high cell turnover. We just constantly have new cells that are being produced here, in migrating up. So the cells that are in the wall are getting worn out. They actually just get ejected into the lumen where they can get digested. They actually get broken down and recycled by the same, in your stomach, you're not just digesting that poor cow. You're also digesting the little poor frontline cells that were lining your intestinal tract that are at the end of their life and get dumped into that corrosive soup as well to just get digested and broken down and reabsorbed. So this high cell turnover is also what keeps things okay. So gastric pits, in addition to being kind of the way that the stuff is coming up from the gastric gland, it's also where you have new cells migrating from the safety of the pit into the front lines to replace the worn out cells that are getting ejected into the lumen to get recycled. So these are three things to keep in mind. It's because it's a problem, right? Your design, the system is this tube through you that's designed to dissolve an animal just like you. So you've got to figure out, how do you actually not just dissolve yourself from the inside? Finally, in this thing, I talked about enteroendocrine cells. So we met hormones when we met epinephrine. Let me just remind you all about hormones, because we got to learn hormones anyway. Hormones, they're basically signaling molecules. Right? Basically you have some cell, you've got some receptor, you have some signaling molecule. Oh, look at him. When it binds the receptor, that activates something. Most of what we saw was in the nervous system, where you actually released a neurotransmitter right into the synapse. right near where you have the target receptor. In hormones, it tends to be more general. These tend to be released into the bloodstream. And then once they're in the bloodstream, they just go everywhere. And anything, any cell anywhere in your body that has the appropriate receptor is going to be able to respond. We saw that with adrenaline, with epinephrine. If your adrenal medulla releases epinephrine into your body, every cell that has has the appropriate adrenergic receptor is going to respond. Your heart will speed up, and your bronchial tubes will dilate, and your pupils will dilate, and whatever, vessels to your muscles will open up. So hormones are signaling molecules typically released into the bloodstream. Sometimes we also talk about paracrines, where you release a hormone and it doesn't even have to go into the bloodstream. It can just diffuse to its neighboring cells and affect them, just because it's still kind of working at a distance, though. It's not like directly released like in a synapse. So people talk paracrines, as opposed to like endocrine, where you release into the bloodstream and it goes everywhere. Paracrine you release and it might just diffuse into just the surrounding fluids and be mainly affecting neighbors. But the idea of hormones, we usually think of these kind of more famous hormones, thyroid hormone or sex hormones. But hormones are used in the control of lots of things in your body. And they are critical in the control of your digestive processes. And the first place people found hormones was trying to understand digestion. So these enteroendocrine cells, this whole big aside here, these enteroendocrine cells, they make hormones that are important to control digestive processes. Like if you're wondering, like, well, what stimulates the cells to make the acid and stuff like that? It's actually hormones. Like one of the important hormones that your stomach makes is called gastrin. This is a hormone. Gastrin is actually going to talk to these guys to secrete all of their stuff. So it's a little non-intuitive that there's going to be this kind of roundabout thing where these cells in here are going to send a message out that then talk to these other cells to do their things. We're going to meet some other important hormones when we get down to the small intestine as well. And that's just a few. There's a whole bunch of hormones involved in control of digestion and in control of satiety, like how much you feel like you need to eat or not eat. There's a whole thing involved there as well. We're not even going to go there. So what triggers those little enteroendocrine cells to secrete gastrin? Tells them, like, we would like you to turn on the gastric secretion and get the acid and proteases going. It turns out the first phase of gastric secretion is called the cephalic phase. And what does cephalic mean? People know? I see Peter's got the head. This is just thinking about food, thinking that you're going to eat, it sends a message. Again, we talked about digestion is promoted by parasympathetic, and the primary parasympathetic nerve is the vagus nerve. The vagus nerve is going to go down and talk to your stomach and trigger these enteroendocrine cells to release gastrin and start the secretion of gastric juices into your stomach. So this is just thinking about the food. This would be what we'd call like a feed forward mechanism, right? It's anticipating a change. You haven't actually eaten yet, but you're gonna be eating it. And so why not start turning the, warming up the system. An interesting thing is this only seems to work if you actually have an appetite for the thing that you're, like if somebody shoved a thing of gruel in front of you and like, eat this. And it's like, okay, I better eat this or else I'm gonna like just die of starvation. Then this isn't gonna work. But if someone put like a, I don't know, a cannoli in front of you or something, you're like, oh my God, I am so stoked to eat this thing. It's definitely gonna turn on the juices before the thing ever passes your lips. Two, there's the gastric phase. So this is when the food actually hits your stomach. What are the things that are going to be happening here that are kind of the stimuli to turn on the gastrin?
Peristalsis turn it on at all?
No. I mean, peristalsis was what brought the food from your throat into the stomach. But what kind of things is going to happen in your stomach for your stomach to know it's probably time to start turning on the juices?
Chemoreceptor?
Yeah, so one thing is going to be chemoreceptors, presence of protein. Exactly. So if there's protein around, that means, oh, it's probably a good idea to start getting these things going. What other things tell us like, oh, there's something in the stomach?
Stretch receptor?
Yeah, totally. Stretch receptors. So you've got stuff like that going on that is helping you, helping control. So the last phase I'll put here, like intestinal phase. This tends to be inhibitory. I'm adding this here. If the small intestine is still working on what it's got, it might send a message back up to the stomach. Like here, we're back here. If here we're in the small intestine, and in the small intestine, you're still working on stuff, and the stomach is squirting more things in, and the small intestine's like, slow down. I'm still working on the last thing you squirted in here. It can actually send hormones that talk to the stomach and slow down gastric emptying. So that's what we mean by this intestinal phase. The intestine sends messages through hormones that tell the stomach slow down because it still needs more time to work on what the small intestine's already got. Foods that are like high in fats take longer to break down and are going to actually make you feel full longer because that's going to cause more of this inhibition of gastric emptying. The small intestine is going to take longer to break down the fats, means they're going to be sending these messages to the stomach saying, slow down and give me more time to work on this stuff. So the kind of food you eat actually plays a role in, how fast the stomach ultimately empties out. Before we leave the stomach, I will just mention, what happens if this protection from digesting yourself breaks down. There's a couple of things that can go wrong here. One is you actually, despite all of this, you actually do start eating into the wall of your stomach. You know, that is called a peptic ulcer. They now are pretty, you know, they know that it's often actually at least involving a bacterial infection as well as Helicobacter pylori. So now it's not just like, oh, you got to relax, don't be as stressed. It's like, take these antibiotics, it's probably going to help. I think, yeah, I've told this story. The guy who first came up with this hypothesis and people thought it was ridiculous, and he was pretty sure he knew what he was onto something, and he just drank a culture of this bacterium and started, like within days, was just full of stomach ulcers. So, you know, it was like the beginning of like, it really is something to explore here. You know, you can actually have perforating ulcers that go all the way through the wall. It's kind of crazy. You can have duodenal ulcers. If this is duodenum, is the beginning of the small intestine. So if you got a hole there. The esophagus we should talk about. You know, the esophagus normally is not going to see digestive juices. You know, it has protection in its epithelium, which is this stratified squamous epithelium, a lot like kind of your normal skin, because it's protected from friction. You're swallowing food going down this esophagus, so you have protection from friction, but not from corrosive like digestive juices. So if this cardiac sphincter here is not holding tight, and you end up getting these digestive juices going back up into the esophagus, you know, then it really hurts, because that's going to actually start burning the lining of the esophagus. You know, that's heartburn. If it's getting up into there, you know, it's right near where your heart is. That's why it's called heartburn. In fact, that part of the stomach is called the cardia. So GERD, they also again call it gastrointestinal, no, gastroesophageal. That's gastroesophageal reflux disease, GERD. You know, that basically, reflux means kind of gurgling back up. So gastroesophageal reflux disease means that your acidic stomach contents are bubbling up into the esophagus that is not designed to handle corrosive acidic goo. And so it starts damaging the mucosa, and it feels really uncomfortable. Obviously, you're more likely to have this if you really stuff your stomach and create more pressure. That's more likely to push the goo up into the esophagus. Obviously, this is more likely if you've eaten a big meal and you're lying down, or even depending which side that you're sleeping on when you go to sleep. Your stomach is here on your left side. And if you're sleeping on that side, it's more likely to be putting pressure on the stomach, shoving, you know, you probably, you know, if you're having like heartburn while you're going to bed because you had a huge meal before you went to bed, sometimes just rolling onto the other side can make a big difference, because there's just less pressure on the stomach, forcing the goo up into the esophagus there. So, ta-da, that's the stomach. And also kind of bringing in a lot of the other things that we wanted to talk about. Now we're going to kind of speed things up a little bit. Stomach is going to dump into the small intestine. The first part of the small intestine is called the duodenum. Duodenum. Stinky. That's just the first part of the small intestine. And it's going to feed down into the main part of the small intestine, which is the jejunum. This is kind of a mixing zone. This is where you have the chyme, the digesting food from the stomach squirting down. It's also where you're going to have the secretions of the pancreas coming in. She kind of comes in from the other side here. The pancreas is going to squirt in all sorts of digestive enzymes. So it's got like has digestive juices. It has amylases. There's a pancreatic amylase, which is going to obviously break down starches. It has proteases. The protease from the pancreas, they call it trypsin instead of pepsin, but it's the same idea. It's going to break proteins into shorter chains of amino acids. It has lipase, which you got to be up close and personal with almost, at least virtually. You got to see pictures of people working with it. Pancreatin. It makes nucleases, which break apart nucleic acids to nucleotide bases and stuff. So that's one part of the pancreas. It has this juice that is full of enzymes that break down anything. That's why I was saying like protein breakdown in the stomach doesn't matter because you've got proteases from the pancreas in the small intestine. It doesn't matter that your salivary amylase gets neutralized by the acid in your stomach because there's pancreatic amylase to continue the job once it gets down into the small intestine. So that's one thing that the pancreas does. The other thing that it has is a bicarbonate juice. Bicarbonate, HCO3 minus, this is the stuff, this is baking soda. This neutralizes acid. So this is basically to neutralize the acid. So the stuff that's coming in from the stomach is very acidic. The bicarbonate juice neutralizes the acid and actually makes the digestive goo alkaline for the rest of its passage through your digestive tract. So that's bicarbonate juice and digestive enzymes from the pancreas. The other thing over here is your liver. Your liver does lots of things. We've already mentioned that the liver is processing nutrients from digestive tract and storing nutrients and interconverting things. We talked about the liver making plasma proteins. Another thing that the liver does is make bile, which ends up getting stored, kind of makes it, but then puts it into this little thing called the gallbladder. The gallbladder stores the bile, stores and concentrates the bile. And then when the digestive tract needs it, there's a little bile duct to squirt it into here. And we saw in lab how bile plus lipase really increases the efficiency of fat breakdown. So the bile, while it doesn't actually, it's not an enzyme, it doesn't actually break things down, it's emulsifying the fats and make them much more easy to break down by the pancreatin. So that is kind of the main things entering here. There's two more hormones that you should know that are controlling secretion by the gallbladder and by the pancreas. So let's put those in here. So hormones released by the small intestine. Again, this initially when people were trying to understand the physiology of digestion, they knew that when food entered the small intestine that the pancreas and the gallbladder started secreting, and they wanted to understand the mechanism. And they cut every single nerve. I think it was in dogs. They would cut every connection they could find between the pancreas and the liver and gallbladder and the small intestine. And still, as soon as the food ended up in the small intestine, the pancreas and the gallbladder would start doing their thing. They're like, how does it know? What's going on? What's the message? And they realized it was hormones. It was these kind of these signaling molecules, which are very hard to detect because they're usually at very low concentrations, getting dumped into the bloodstream, traveling through the bloodstream, and then being detected by receptors, at the pancreas, at the gallbladder. So one of the hormones released by the small intestine is called cholecystokinin. Although if I ask about this on a test, you can just call it CCK. That's what people usually call it. CCK basically triggers bile secretion or bile delivery from the gallbladder. It basically causes the gallbladder to contract and squeeze bile down, and enzyme juice from the pancreas. So cholecystokinin is basically going to be triggered when there's fats and other nutrients that show up and says to the pancreas and to the gallbladder, oh, we got some stuff to digest here. Please give us the enzymatic tools we need to accomplish breakdown, as well as the emulsifier that will help make our fat breakdown more efficient. The other hormone that you should know is called secretin. Tells pancreas to deliver bicarbonate juice. So now that I've got this again, cholecystokinin, also known as CCK, secretin. These are the two hormones that you should know, in addition to gastrin, which we talked about from the stomach. Cholecystokinin is going to tell the gallbladder and the pancreas we want bile and digestive juices or enzyme juices to break stuff down. Secretin tells the pancreas we need more bicarbonate juice. What's going to be the stimulus for secretin?
pH.
Exactly. Low pH is going to trigger the release of secretin, in which case the pancreas will deliver more of this alkaline bicarbonate juice to neutralize that acid. So that we've got that going for us in our digestive tract. So for the next part, we've gone, we're going to go into the rest of the digestive tract. We had the duodenum, jejunum, ileum. So jejunum, this is again the main site of breakdown and absorption. Ileum is more about reabsorbed bile salts. Right, we're dumping in bile salts from the gallbladder here at the top. And we're breaking down the fats, but then it's like, why poop them out? Why not just pull them back into your body, bring them back up, and the liver can then reassemble and put them back in. So the bile salts are not just getting made from scratch and pooped out all along the way. They actually get made and dumped in at the top of the digestive tract, but then reabsorbed at the bottom of the intestine to get reused. So it's more of a kind of merry-go-round for those guys rather than a one-way trip. Jejunum, main site of breakdown and absorption. This is what we're going to focus on now. Again, breakdown we've already dealt with. The breakdown is occurring with all those digestive enzymes from the pancreas, the bile from the gallbladder. For absorption, we're going to have to bring the nutrients across the mucosa into the bloodstream. And if you want to increase, when we talked about diffusion rate, if you remember diffusion rate, a big part of it was surface area. So we're going to want to have a really high surface area. If we want to like pull stuff across into our body, we're going to want to have a really high surface area to pull stuff across. And you don't need to know the details of this, but we can do at least, I can show you the basics of this in the PowerPoint from the text, which is going to be way faster than me just trying to draw it. There we go. So the basic idea, your small intestine has a, we need to do that. Let me make a little bit of a darker color. This is the small intestine here. If you cut it open, it's got all of these are called circular folds. And then if you look more closely, it actually looks kind of like a shag rug when you're looking at the surface. All these, they're called villi, little finger-like projections of the mucosa going up and down like little fingers. And then if you look at the individual cells that make up the epithelium, the columnar cells, they have microvilli, which we call the brush border. So you've got these like 3 layers of folding. You have these circular folds, which are really big. You can actually see them. They're like a 1/4 inch. Then you have the villi, which you can see because it looks like velvet. Kind of like, velvet doesn't look smooth. It looks like it's got some kind of texture. Microvilli, you can't even see without an electron microscope. But you end up getting a crazy surface area. So all these serve to really increase the surface area. Things that I've read are if you unfolded every little fold here, you'd have a surface area about the size of a football field. So you've got a crazy amount of surface area to absorb nutrients from the lumen into your bloodstream. So this is kind of just showing, these are the villi kind of going up, increasing all the surface area. Down here in this loose connective tissue, we call the lamina propria. This is where you're going to have the capillaries that are going to actually be where the sugars and whatever. This is transepithelial transport is going to be happening right here. Let me get a darker thing that you can actually see. You're going to have sugar here. It's going to get pulled across, absorbed into the bloodstream, and then carried into the hepatic portal system to your liver. So, and let's see, this brush border, again, each of these cells here has this crazy folding just of the cell membranes called the microvilli, which we call the brush border. The brush border will be where we find the last stages of digestion. Let's not get into any of that. All right, so what we're going to do now is we're going to focus in one of these little finger-like projections and just look really briefly at the absorption of nutrients happening there. We're okay here. This is basically the lumen of your intestine. This is the mucosa here, the simple columnar epithelium. Then we are going to have our little capillaries. So the reason we can do this really fast is because you know it already. You're just going to use, for proteins, for carbohydrates, it's just going to be transepithelial transport. You'll have some glucose, and the glucose is going to come in a symport with sodium, and then it'd be a carrier on the other side and come out the other side and then get absorbed into the bloodstream. So if you're looking at amino acids or monosaccharides, it just happens kind of the way you've already been familiar. The place where we have to have a different story is with the fats. Fats do not dissolve in water. So we need to, I've mentioned before the idea of lipoproteins, but I will go a little deeper now. Basically, it's lipid, there's kind of fat, which normally would not dissolve in water, but then you complex it with a protein part that is more on the outside. And now we have something that can dissolve in water and actually can act as a transportation vessel for the lipid. So this is how fats are carried about in your body, is complexed into these lipoproteins. There are 3 lipoproteins you need to know. If you have one that has not that much, because fat floats. If you have more protein and less lipid, that would be like a high density lipoprotein. HDL, high density lipoprotein. If you have more fat and less protein, that would be low density. That would be what we'd call and LDL. In general, the LDLs tend to be more involved with depositing fats into your body. The HDLs tend to be more with taking fats away for disposal. When people often talk about the good fats and the bad fats when they're doing a blood workup, the LDLs are the quote unquote bad fats and the HDLs are the quote unquote good fats. Obviously, you need both of them. They're both doing important things, but the ratio, there is kind of optimal ratios. If you have like an overabundance of depositing more fats into your tissues compared to taking stuff away, that's usually not a good sign. So, you know, they look at the ratio of the HDLs and the LDLs in your blood to kind of get a sense of your overall kind of fat metabolism at the moment. The other lipoprotein that we need to know to finish our absorption of fats from the digestive tract are specialized ones called chylomicrons. They are just lipoproteins that are specifically involved in this whole absorption of fat. The basic idea here is, whoops, I need that. But the basic idea is you're breaking down fats. The little fat breakdown products get taken up, but they get assembled into these little chylomicrons, these little lipoproteins by these intestinal endothelial cells. They call these like enterocytes. It doesn't matter. But basically, we're making little lipoproteins. So for lipids, assemble breakdown products into these chylomicrons. So I'm taking the lipids and I am assembling them into these little lipoproteins, which can float around in water and carry the fat around, but they're big. They're too big to fit across the wall of a capillary. So you can't actually absorb them directly into the capillary. So how is it that we're going to absorb them into the body if they can't get into a capillary?
Lymphatic system.
Exactly. So we are going to have lymph capillaries that are coming up in here. Let me like erase this other stuff now. So this is a lymph capillary. We call them lacteals because when the fats get into there, it looks a lot like milk. That's why it's called lacteal. But basically, these have those kind of flappy endothelial cells that actually can allow the chylomicrons to get into the lymphatics. And then where do they go then? Where do they end up then?
Hepatic portal system.
So they don't. So this, in fact, that's going to be something that's different about absorbing fats than absorbing all the other nutrients. Like, where do all of the lymphatic vessels ultimately drain? Say it again, Allison.
Is it the vena cava?
Yeah, exactly. They just go dump back into the vena cava and back into general circulation. So those fats are going to end up in your circulatory system, but it's by a roundabout path. They get into the lymphatics, but all the lymph ultimately drains back into your closed circulation. But kind of back to Anne's thing, it's actually not going to the hepatic portal system. So that actually can be, that can actually be relevant sometimes, like things that are absorbed, fats and things that are absorbed as part of fats actually can be a little more dangerous sometimes because they don't necessarily get detoxified going through the liver on the first pass. So it actually sometimes is relevant to know that lipids don't go through the liver. They get dumped straight into the bloodstream. So that's it for the small intestine. You should know the difference between absorbing lipids versus absorbing everything else. And then we're going to finish up really quick. Large intestine. So your colon here. Like I said, it's a bunch about water reabsorption. Because if again, not enough water reabsorption, people can die of diarrhea even because, you get super dehydrated. There's also a bunch of bacteria in here. The bacteria can break down stuff and make some vitamins that you can absorb. There's like some B vitamins, K vitamins that you actually can absorb that are made there. The bacteria also break down things you can't break down, like cellulose, and make gas, right? Again, things that are more cellulose, like beans, are going to make you toot louder, right? So luckily we have the rectum. The rectum has these kind of valves that help separate flatus from feces. Right, you want to be able to have the gas escape without having like spraying feces out the anus there. So the rectum actually allows kind of the gas to get out without having the feces. There's two sphincters, just like we had in the urinary system. There's going to be an internal and an external sphincter. I'll just type it super quick to make life easier here. Internal involuntary sphincter. And the external voluntary anal sphincter. Again, the internal sphincter is smooth muscle. It's going to get triggered by a reflex as the rectum stretches out and the lower part of the colon here stretches out. This guy will relax, which will then put pressure down on the lower part here. You'll feel the urge to defecate. And then you can make a choice. It's like this appropriate time and place to relax the external voluntary anal sphincter and poop. What else do we want to say about this? These bacteria, question?
Wait, what does it say on the right? Separate flat.
Oh, flatus.
No. Thank you.
Flatus is the kind of fancy word for like gas. There's lots of fun words. Another borborygmus. Plural is borborygmi. It's another one you should probably know. Does anyone know what borborygmi is or are? Or these are sounds. So you're constantly, if you put a stethoscope, you'll hear all sorts of blah, 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 blah. Because everything's getting mixed, all that segmentation, you're stirring things up. But borborygmi are sounds audible at a distance that are being generated. It's like when your stomach is growling. That would be an example of borborygmus, is like that growl. So it sounds generated by the digestive system that are audible at a distance. What's the fancy word for if gas escapes out your esophagus? What is that? If I wanted to be sophisticated, what did I just do? Or as sophisticated as I could be. Eructation is the fancy word for burping or belching. So eructate. It sounds like, orate, or this eructate is. So anyway, the bacteria are really interesting too, but we're not going to get into them in the interest of time. But half of your poop is actually bacteria. It's not waste. You think about your poop being the stuff you didn't digest, and some of that is true, obviously. You see like the little piece of the corn or whatever. But half of the volume or so is the bacteria. And they're necessary. I mean, if you don't have the appropriate bacteria, it messes with the development of your immune system. You don't even have normal neural development. The bacteria make all sorts of stuff that actually gets absorbed into your bloodstream and can actually have all sorts of effects on different parts of your body. So they're definitely not just kind of totally benign, or not benign is not the word, but they're active. They're not totally passive. They've done these really fascinating studies where they'll take the bacteria from the gut of one rat and transplant it into the gut of another rat, and the two rats will switch their baseline weights even though they're not changing anything that they're eating. So there's something about the bacteria interacting with their basic metabolism that changes the way they metabolize their food. They can change what kind of food you crave. Supposedly, bacteria can actually influence you to want to eat the kind of foods that are the substrates that those bacteria like to break down. So they can kind of play you a little to like eat the stuff that they want to see coming down the pipe. So they're fascinating. What else? What else? I should say just fiber really quickly, just because I'd be remiss not to mention fiber. This is basically like cellulose, which we talked about in the first week of class. Cellulose, which we cannot break down. So that means it makes it all the way down here as still being cellulose, being this polysaccharide. It's the roughage. This is the roughage, and it's the cell walls of plants. And what's nice is it gives some bulk. The stuff here that's in your large intestine has some bulk still to it. It's not just pure goo. So the muscle can keep it moving along and eventually get it out the other side. Again, if things don't move along, you withdraw too much water, that's where you get constipation. So you need to have enough kind of bulk that there's something to actually push along with the peristalsis. So this fiber helps give you some bulk to keep things moving. It also is kind of hygroscopic, kind of keeps some of the water in, keeps you from getting it totally dried out, and like I said, turning into a rock. So there's like some science behind fiber is good for you or keeps you regular. It definitely does. Okay.